Alright, welcome back. In this video, we're going to go over glycogenesis. So I know I've been gone for quite a while. I had spring break, so I decided to, relax a little bit. But I also started a new lab position, so I'm dealing with mice and doing drug addiction studies. So that's a brand new thing I'm doing, so I wanted to, focus a little on that. But I'm back, I promise, I'm back. The videos are back. So we're going to go over how to make glycogen in this video, and we're going to go over through all the pathways and whatnot. It sounds like gluconeogenesis, right? Glycogenesis and gluconeogenesis. We're not making glucose. We're making glycogen. So let's start off with going over why we're doing this. What's the purpose? Why make glycogen? So glycogen is a way to store excess glucose when we don't need it. So let's give like a real life example here. So say if you were to go eat a big meal, say like 2,500 calories or maybe 3,000 calories. I know it's a little excessive. But say you go eat a big meal. And then you go home and you rest the entire day. You don't do any physical activity whatsoever. Well, now you have excess glucose because we're not burning enough calories, right? We're resting. We're not burning enough calories. So excess glucose is going to be turned into glycogen as a temporary storage site. Now, excess glycogen has a cap. We can only store around, I think it's around like 2,000 calories of glycogen. If we eat more than that, and we need to store more glucose than 2,000 calories, we're gonna first use up our 2,000 calorie glycogen supply, and if we still need to store more glucose, then the rest will be turned into fat. So this is the way, to avoid body fat. If you don't want to gain fat, you need to exercise. Now, a lot of people think glycogen's bad. It isn't. It's actually keeping you alive. Because what happens if you were to have a really busy day, for example, right? You go to class, we're called students, right? We go to class, and then, we're in class for like 2 hours, 3 hours, and you have another class to go to, you have homework to do, or, whatever, study, where you don't have time to eat, or you have a job, right? You don't have time to eat. Well, good thing we have glycogen, because the excess glycogen we have stored will be used as energy, so you don't have to eat. Okay, so it's kind of like a starvation mechanism, or hunger mechanism in a way. So this is kind of saving you, so you can actually function and do your daily activities without eating. Of course, it's a 2,000 calorie cap, right? Once you're out of those, then you start burning fat, and that's how to burn fat. Okay, you have to first burn through your glycogen storage, and then you can start burning fat. So, glycogen is stored mainly in two places, the liver and skeletal muscle. There's differences. So, there's no way to actually tell where it's going to be stored at. It's just random. However, they do two different things. If glycogen is stored in the liver, it's actually be able to be transported to different areas of the body wherever it is needed. So if your body needs energy, say in your kidneys, for example, or your brain, the glycogen will actually rip off and be transported to those organs for energy consumption. That's the cool thing about the liver and glycogen storage in the liver. If it's going to the skeletal muscles, if glycogen is stored in the skeletal muscles, it only has one duty. It can only do one thing. It can only be used for skeletal muscle use, so for physical activity. It cannot be transported. Once it gets to the skeletal muscle, it's not leaving. It cannot leave. It can only be used for physical activity. So, another way to put it actually is that, for example, if you go on a run, and you start feeling that burning feeling in your legs because you're running out of energy, it's going to start burning glycogen. That's your body's way of telling you, oh, I'm burning glycogen, I'm out of energy. That's that burning feeling. Okay? So step one. Step one is actually very easy. It's glycolysis. It's the first stage of glycolysis, the first step of glycolysis. We take glucose, we're starting with glucose. We have an ATP molecule. That ATP molecule is going to strip away one phosphate, and that phosphate group is going to be attached to carbon number 6 using hexokinase. So pretty easy. It's the first step. Step 2. Step 2, we're going to use an enzyme called phosphoglucomutase. And think about mutase as a mutation. we're mutating this molecule. All we're doing here is moving the phosphate group from carbon number 6 to carbon number 1. It needs to be on carbon number 1 for this to happen. Okay, so this is a way to confirm that, okay, we are going into glycogenesis. Pretty simple, right? We're halfway done. Step 3 is when we get a little complicated, and we're going to take it very slow. It is complicated. So we have our glucose 1-phosphate molecule here we just created from the last step. We're going to get a random UTP molecule. Okay, so you know how we have like ATP, right? Adenine triphosphate, then we have like GTP, guanine triphosphate. Now we have UTP, uridine triphosphate. So it comes from, it's a derivative from uracil, so like RNA. So isn't that pretty cool where these things come from, like DNA and RNA from nucleotides? So we have a uridine triphosphate. So uridine has 3 phosphates on it. We are going to take one of those phosphates and attach it to glucose 1-phosphate, okay? And it's going to create UDP glucose, uridine diphosphate. And we're going to use the enzyme UDP glucose pyrophosphorylase. Now you're probably thinking, why is the final molecule called UDP? Shouldn't it be UMP glucose? Because we only stripped away 1 phosphate from UTP, right? 1 phosphate meaning monophosphate. We stripped away 1 phosphate from UTP. So shouldn't it form UMP glucose? I see that logic, I totally do. But the reason it's called UDP glucose is because we already have a phosphate group on carbon number 1. So the molecule already has 1 phosphate. Then we take the UTP molecule, we shred off one phosphate from that and attach it to the glucose 1-phosphate. So now we have 2 phosphates, and that's why it's called UDP glucose, uridine diphosphate glucose. So that's the first half of this. The second half is a little bit complicated. It's not too bad, but let me explain like this. So let me draw it. So here, we just used the UTP, right? And it became UDP, okay? Because one of the phosphates, 1 phosphate, went here, okay? Now we're left with UDP. This, we call this, it's a fancy name actually, we call this pyrophosphate. We call this pyrophosphate. And this is actually abbreviated as PPI. Okay? We call this PPI. Now here's what we're going to do. We're going to use an enzyme called inorganic pyrophosphatase. And we're going to basically turn this PPI or pyrophosphate into 2 inorganic phosphate groups. Okay? We have now created 2 phosphate groups. This is important because this drives the original reaction forward. It releases so much energy, a lot of energy. So this is like a coupling reaction, what we talked about previously in this course. So releasing these 2 inorganic phosphate molecules drives this entire reaction forward. And it's irreversible. We cannot go back from this. So once we create UDP glucose, it is destined to become glycogen. That's it. We're done. So honestly, that's the hard part of it, right? If you can get this, you basically know glycogenesis. So the last step is we're going to take the UDP glucose molecule we have just had, and we're going to combine it with an original existing glycogen molecule. So it needs a primer for this to actually happen. So to actually add on to glycogen and make more glycogen, we need a pre-existing glycogen molecule. So it's called a primer. So the primer can exist as in a glycogen molecule that already exists, or glycogen. It's a protein. So all you have to really need to know here is that to make glycogen, you need glycogen to exist in the first place. I know it's a little complicated, but we always have some glycogen present in our liver or skeletal muscle. There's always some. So to actually add on to it, we need it to exist, right? We need UDP, glucose, to exist with the glycogen molecule, a primer. So what's going to happen is we're going to use the enzyme glycogen synthase, and the UDP that's attached to the glucose will shred off. It's going to leave, okay? It's going to leave. And so the glucose will join onto the glycogen molecule, and we're going to create +1 on the glycogen chain. So let me draw it out for you. So here we have, say, the glycogen molecule that already exists, right? It's already in our liver. All we just did is add the glucose molecule onto it, right, by shredding off the UDP using a glycogen synthase, and it becomes part of the glycogen strand now, or it's a chain. And that's how we create glycogen. So we ended with glycogen. And that's basically it. It's pretty easy. So if you found this video helpful, please like and subscribe. Until next time, later.